Good day, this is Professor Will, CBMD PhD. Today is Monday, April 12, 2021. It is 10:35 and 35 seconds p.m. Eastern Standard Time. Today's research, Neanderthal ancestry. So let's hop to it here. Neanderthal ancestry identifies oldest modern human genome. Date April 7, 2021. Source, Max Planck Institute for the Science of Human History. Summary. The fossil skull of a woman in Czechia has provided the oldest modern human genome yet reconstructed, representing a population that formed before the ancestors of present-day Europeans and Asians split apart. The segments of the Neanderthal DNA in its genome were longer than those of the UST Ishim, individual from Siberia, the previous oldest modern human sequence, suggesting modern humans lived in the heart of Europe more than 45,000 years ago. Ancient DNA from Neanderthals and early modern humans has recently shown that the groups likely interbred somewhere in the Near East after modern humans left Africa some 50,000 years ago. As a result, all people outside Africa carry around 2% to 3% Neanderthal DNA. In modern human genomes, those Neanderthal DNA segments become increasingly shorter. Over time in their length can be used to estimate when an individual lived. Archaeological data published last year furthermore suggests that modern humans were already present in southeastern Europe 47 to 40,000 years ago. But due to a scarcity of fairly complete human fossils and a lack of genomic DNA, there is little understanding of who these early human colonists were, or of their relationships to ancient and present-day human groups. In a study published in Nature, Ecology and Evolution, an international team of researchers reports what is likely the oldest reconstructed modern human genome to date. First discovered in Czechia, the woman known to researchers as the Golden Horse in Czech displayed longer stretches of Neanderthal DNA than the 45,000-year-old UST Ishim, individual from Siberia. The so far oldest modern human genome analysis suggests that she was part of the population that formed before the populations that gave rise to present-day Europeans and Asians split apart. A recent anthropological study, based on the shape of the skull, showed similarities with people who lived in Europe before the last glacial maximum, at least 30,000 years ago. When radiocarbon dating produced sporadic results, some as recent as 15,000 years ago, the researchers found evidence of cow DNA contamination in the analyzed bone, which suggests that a bovine-based glue used in the past to consolidate the skull was returning radiocarbon dates younger than the fossil true age. However, it was the Neanderthal DNA that led the team to their major conclusion about the age of the fossil. Prehistoric conclusions carried about the same amount of Neanderthal DNA in her genome as UST Ishim or other modern humans outside Africa, but the segments with Neanderthal ancestry were on average much longer. The results of our DNA analysis show that she lived closer in time to the admixture events with Neanderthals. The scientists were able to estimate that she lived approximately 2,000 years after the last admixture. Based on these findings, the team argues that she represents the oldest human genome to date, roughly the same age as, if not a few hundred years older, than UST Ishim. It is quite intriguing that the earliest modern humans in Europe ultimately didn't succeed, just as with UST Ishim and the so far oldest European skull. From OACE-1, hers show no genetic continuity with modern humans that lived in Europe after 40,000 years ago. One possible explanation for the discontinuity is a companion ignimbrite volcanic eruption roughly 39,000 years ago, which severely affected climate in the northern hemisphere and may have reduced the survival chances of Neanderthals and early modern humans in large parts of Ice Age Europe. As advances in ancient DNA reveal more about the story of our species, future genetic studies of other European individuals will help to reconstruct the history and decline of the first modern humans to expand out of Africa and into Eurasia before the formation of modern-day non-African populations. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video lecture was created for educational purposes only. I hope you enjoyed listening. Until next time, have an awesome day.